So I again start by thanking Oncostem and Canasis Breast for sponsoring this program. Next. So attitude determines the altitude. So how big you become in life, how well you grow, it depends on your attitude. And again, if you have a know-it-all attitude, you are not going to be here attending this course. So I thank you all very much, all 93 of you, for having taken the time off to be part of this program, which I think is very, very important for all oncologists. Next slide. So let's start with the title, Novel Biomarker. So what is a biomarker? I'm sure that all of us say casually we know it, but let us look at what is the clear definition of what a biomarker is. So a biomarker is a characteristic that is objectively measured and evaluated as an indicator of three things. It could be one of three things. It could be more than one thing. Either it is a normal biological process, or it is a pathogenic process, or it's a pharmacological response. And all these three are applicable to oncology. For oncologists, it is very common to look at a biomarker like total WBC count and platelet count, right? Because we want to know whether the patient is fit to receive whatever we want to do for the patient or not. In addition, cancer specific biomarkers that we have been using for ages, like CA125 for women for ovarian cancer and PSA for men for prostate cancer. But today, really, for personalized oncology, our focus is on biomarkers like PDL1 and HER2 gene and so on and so forth. So various types of molecules, which could be DNA or a protein or a hormone, can serve as a biomarker for health as well as in illness indicator. And that is why novel biomarkers are really important for personalized oncology. Next slide. Immunohistochemistry, all of us know, but this is really important. So we must pay some attention just as a revision. This is a peroxidized staining, and therefore any brown pigment that you see is a positive for immunohistochemistry, irrespective of what monoclonal antibody is used. On the top left-hand side, you will see only blue color and no brown color. That means the score is 0. On the bottom left-hand side, you start seeing some outline on the cell membrane, which is a score of 1 plus. On the top right-hand side is a score of 2 plus because the brown is in larger number of cells as well as it is more intensity in staining. And 3 plus is what you see on the bottom right-hand side. So immunohistochemistry continues to be a biomarker of sustained interest because of HER2 and anti-HER2 therapy. And there is a resurging focus after ALK became positive for targetable personalized therapy in lung cancer, and now, of course, with PDL1. Next slide. So now we come to the terminology of companion diagnostics. When immunohistochemistry is done and a specific monoclonal antibody is used, this is what is enforced by regulatory authorities. So US FDA and EMEA says that if I have a patient of melanoma or non-small cell lung cancer and I want to use pembrolizumab, then a PDL1 should be tested by a monoclonal antibody called 22C3. And this is the companion diagnosis. If I want to use nivolumab in the same patient, then the PDL1 should be expression should be done with a monoclonal antibody which is different, that is 28-8. And if I want to use durvalumab for renal cell carcinoma, then I must use the monoclonal antibody SP142, which also looks for PDL1 expression. Now, another terminology comes into the picture when we talk about atezolizumab. And that is that now the cell or organ of origin of the cancer does not matter. It is a tissue agnostic biomarker companion diagnostic. And if I use SP263 monoclonal antibodies and it is positive for PDL1 expression, irrespective of what cancer it is, I can use atezolizumab for these patients. Next slide. So let's also refresh our memory about human genome and DNA. And again, this is a revision. All of you really know of it. An exon is a portion of the DNA that codes for amino acid. So from DNA, the message goes to RNA and then a protein production occurs. And it's a sequence of amino acids in the protein that determine what is the output. Intron is a stretch of non-coding DNA found between exons, and these intervening sequences can be really long. And a gene is a combination of exons and introns that have a specific function. So that is how gene is formed. If you look at all the exons in the human genome, they form what is called an exome, and this constitutes 1% of the human genome. Now, this difference between genome and exome is vital because when we do next generation sequencing, if we want to reduce the cost, if we want to reduce the time and make it a more efficient process, we might want to focus only on exons and then we do an whole exome analysis as opposed to a whole genome analysis. Next slide. Now, this is the traditional terminology for what happens with DNA. So, we have a mutation, which is a change in the structure that causes disease. There could be a polymorphism, that is a change which is found at a frequency of 1% or higher, but we still don't know whether it causes a disease or not. And many of them are single nucleotide polymorphism. So, this is the traditional terminology that we have been using. And based on this terminology, next slide, we could have different types of mutations. The first one here is the point mutation. On the left hand side is the TAC versus ATG, which is the normal protein, and this is called wild type. There are no mutations. If the C is replaced by T and the G is replaced by A, then you have a change in the DNA structure, but the protein, the amino acid sequence remains the same, and therefore this is known as a silent mutation. It has no implications as far as cancer is concerned. The third one is where the C is the first T is replaced by A and the first A is replaced by T. So tyrosine is replaced by asparagine. A new amino acid comes into the picture. This leads to a production of a protein that is faulty, and this is known as a missense mutation. The last one is where the C is replaced by G and the G is replaced by C, and this results in a stop codon. So this leads to an incomplete protein production, and this is called nonsense mutation. So these are the three types of point mutation. Next slide. But the changes in the structure of the DNA could be much larger, as is shown in this slide. On the first one is a large chunk of the DNA that has gone out, gene that has gone out, and that is called deletion. In the second one, you can see the example where the blue box represents the gene that is duplicated, and this is classical of HER2 amplification, and this duplication could be not only 2 but 20 or 30 times. The third example is an inversion, whereas the direction of the coding DNA is reversed or flipped. The third one is an insertion, whereas a part of a chromosome is taken out from chromosome 4 and is inserted into chromosome 20. There is a translocation where the insertion is on both the chromosomes. So there is one part that goes from chromosome 4 to chromosome 20, and the reciprocal part from chromosome 20 comes back to chromosome 4. So that is a translocation. And classically, what you see in CML in chronic phase between 9 and 22 is what is called as a balanced reciprocal translocation. What that means is that not a single nucleotide in the DNA is missed or lost. It is totally balanced what goes in and what goes out. Next slide. And finally, we have something called a frame shift mutation. And this frame shift mutation means that there is either an insertion or a deletion of nucleotides which are not in multiples of 3. If it is in multiples of 3, it is changing the sequence of an amino acid. Here, these, because it is not multiples of things, this is the most detrimental mutation that you can see, and that is called frame shift mutation. It can either alter the amino acid sequence or totally stop the production of that protein. Next slide. It could also be terminology like synonymous mutations, which does not change, result in change in amino acid sequence, or a non-synonymous mutation. And more important, which all of us are familiar with, is the difference between a germline and a somatic mutation. So germline mutation occurs in body's reproductive cells, which are either the ovum or the sperm, and therefore this defective DNA will then be passed on to the each and every cell of the offspring. For instance, the BRCA gene mutation. The somatic mutation occurs in cells other than the cells of the sperms and the ovum, and therefore do not pass on to children. And this is just a sharing the terminology again, which you are already aware of. Next slide. So DNA variations could also be gene copy number, as we have already shared with you about HER2 amplification. And structured variation, therefore, is a combination of mutations, all the three types of mutations that I mentioned earlier, plus copy number variation. What is very important for us to understand is that sometimes these mutations, we don't really know what is the significance as far as the treatment or the clinical implications are concerned. So there could be a genetic variant which has no or unknown relationship with the disease, and that is known as a VUS, or a variant of unknown significance. And we will talk a little bit later about that. Next slide. So oncogene is a mutation in the normal DNA. Normal gene gets converted into an altered or mutated gene, and this is the first step in the development of cancer. If this gene mutation plays a key role either in the development or the survival or the spread of the cancer, it is known as a driver mutation. And this driver mutation can change as the disease progresses because the patient might have more mutations. Another important thing is a non-mutational epigenetic change. This is a heritable change in the DNA structure that does not alter the underlying DNA sequence. So we can't call it a mutation, but DNA methylation will change something that will affect the progeny. And this is epigenetic change. Next slide. Coming back to the four machines that I shared with you earlier about the next generation sequencing. Next. So what is sequencing? Sequencing could be whole genome sequencing or whole exome sequencing. Whole exome sequencing is 1% of whole genome sequencing. And it is a procedure that determines the sequence of the entire genome or the protein coding region of an individual or a human being. And when we are doing this, we are dealing with millions of base pairs. So the terminology that we are using for this is megabases, which amounts to 1 million base pairs. Next slide. So initially we started with Sanger sequencing, and now we have moved to high throughput sequencing, which is called next generation sequencing. It is rapid sequencing of large portions of the genome, and the value of that is for speed, depth, efficiency, automations, and technology. There are two other terminology that we should understand for NGS. One is sequence coverage or depth of coverage, and that is the number of times an area of interest, a gene of interest, or an exon of interest is crossed while doing the sequencing. It could be 1x, it could be 100x, it could be 5000x. And the greater the depth of the coverage, the more sensitive is the input that we get. The other is the read length, and that is the length of the DNA copy that is generated when it crosses the area of interest. And these read lengths could be longer, which will able to identify more larger mutations as opposed to point mutations. Next slide. This is a comparison of the four technologies with the machines and what happens as far as the read length, the run time, and the daily throughput is concerned. The last column is the approximate cost per 1 GB of the sequencing that happens with this technique. So the Sanger sequencing, which is the traditional sequencing, will cost $8 million per 1 GB of output. Therefore, we are using sequence by synthesis or color spaces. Sequence by synthesis, the example is Illumina HiSeq, which will have a read length of 100 paired ends. It takes about 11 days. It provides the daily throughput of 55 GB, and it costs only US dollars 47. So you can see the difference in the cost. You can see the difference of the labor and the intensity and the reagents required while doing the sequencing. Next slide. And what happens after the wet lab is the huge amount of data that comes out, and that is why it requires bioinformatics. Next slide. TAT or turnaround time for NGS is really high because the intense amount of data that needs to be crunched to analyze, to evaluate before giving out the result. And again, I've shown you that based on the coverage or the read length, the amount of data that needs to be generated is really high. Next slide. Now, when we get a report, it can show something like microsatellite instability or tumor mutation burden, both of which will indicate that these patients might respond better to certain types of therapy. Next slide. And this is a report that has come out from comprehensive tumor profiling from one of my patients. You can see that the tumor mutation burden has been identified by sequencing, which shows that it is high. And this is measured by the number of mutations by MB or megabases of the DNA. And in this instance, it was 36. You can see that AR was defined by immunohistochemistry, and it was a 2 plus positivity in 95% of cells. And there's a P10 mutation that was identified by sequencing of the DNA, and there are two pathogenic variants. One is the variant in exon 5, and the other is the variant in exon 7. Now, when I get a report like this, based on this terminology, how do I interpret this? Next slide. What I can do is I go to my laptop, connect to the internet, go to this My Cancer Genome website. Free access, absolutely no need to pay or ask anybody. I put the P10 exon 7p.k257fs, and promptly it gives out the response that this affects the PIK3 AKT mTOR pathway. It is being addressed by 118 clinical trials, and it is related to pancreatic adenocarcinoma. So I don't need to remember everything. I only need to understand that these are the publicly available databases that I can access to identify what I need to do for this patient. Next slide. This is another example where mismatch repair by immunohistochemistry and MSI by sequencing, microsatellite instability by sequencing has been identified. Mismatch repair is deficient, MSI is high, and therefore it is clearly level 1 evidence that this patient will benefit from pembrolizumab. So if you have a clear-cut report like this, our decision in treatment for this patient becomes crystal clear. But this is possible only if we are familiar with the terminology that is available today. Next slide. And sometimes we have the variations of unknown significance, and then we need to reclassify them whether they are pathogenic or benign. Next slide. So we look at the new terminology that has been approved by the Human Genome Variation Society, and that includes sequence variants, alterations, and allelic variants. Next slide. And this is also approved by three other societies called Human Genome Variation Society, Genome Varium Project, and Human Genome Organization. This applies to DNA, RNA, and protein. Next slide. And this is how the standard variant is described. So there is a prefix, position, and a change. So we have C.4375 C to T in this example. C indicates the coding DNA reference sequence. The 4375 represents the position of the nucleotide affected, and the change in the amino acid sequence is from C to T, nucleotide sequence. Next slide. And this is how this nomenclature is now followed in the reports that we get. And based on this reports, we can interpret the reports ourselves by looking at publicly available databases and verify whether what the report pathology lab is saying is true or not. Next slide. So these are the variant nomenclatures where you can see that DEL, DUP, INS have been added to talk about deletions, duplication, and insertion. I'm running out of time and therefore I have to move a little faster. Next slide. And why is this important? New information becomes available all the time, which is added to public databases every week, every month, every 3 months. VUS therefore then gets assigned a relationship, whether it's benign or pathogenic, and I can interrogate this database at regular intervals for my specific patients without having to take the help from anybody else. Next slide. So when I send a sample to the lab, I insist, I will just take 30 seconds more. I insist that we get the raw data on a pen drive. This raw data allows me to check whether it is a VUS converted to a pathogenic variant or a VUS that becomes benign, and therefore I can guide my patient better without having to repeat the test or pay extra for it. Next slide. Finally, I would like to end by showing this as my last slide, that there is something online, a web portal that is called BRCA Exchange. Like we have Sensex for exchanging stock, this is exchange of available global information on BRCA1 and 2. There are more than 20,000 such variants that have been identified. There are 7,500 with expert classification through Enigma. And this is a data that has been collected from 300 members across 35 countries. With this, I will stop this terminology module, and we can go to the final MCQs, the same MCQs that were asked earlier. But before that, we can take the question answers. So you can project the questions from the audience if possible. We can unmute Anuradha. She wanted to ask earlier, but we could not take it at that time. So this question is from Chandrakanth. Why don't you moderate?
Yeah. So this is a question from Dr. Amishwara to Parikh especially. So yes, the technology has created an epidemic on fear of missing out. So instead of understanding subset biology, so we want to use personalized medicine in all patients. So will chemotherapy also become a part of personal medicine, or it will become redundant like a pager as you showed in your slide? So it is specifically for you.
Right, right. So fear of missing out is always there. No matter how senior or junior you are, people always have the fear of missing out. Either it is a fear of missing out on patients, or fear of missing out on quality of life with family, or fear of missing out for going for a social gathering. Of course, in COVID pandemic, we are not talking about social gathering, but definitely we have to use this information judiciously. And I think the whole purpose of having this certificate course is to make sure that you don't start doing novel biomarker testing for each and every patient. For instance, if I have a lung cancer patient who is not affording anything beyond the first generation TKIs, I would not do any other test except EGFR mutation. But that has to be judiciously used. And in that sense, we will never stop using chemotherapy, because even today, there is a significant portion of patients, for instance, taking the example of lung cancer, where we don't know what the driver mutation is. But the portion of patients who will receive chemotherapy may go down as we go ahead in the future.
Okay, sir. So next question. Yeah. So I was just asking, is there a difference in the amount of tissue required for a multiple test? For example, ALK we do by AHC, PCR we do for EGFR, and Sanger we use for sequencing versus an NGS. So if Anuradha ma'am was there, I was just trying to ask
Yeah,
her
so
specifically.
Anuradha is here, so please
Yeah.
answer Anuradha.
Yeah. Is there a difference in the amount of tissue required?
Tissue. Sorry, that's the typo.
Tissue
Yeah.
required for Sanger PCR, yes, versus doing AGS, yes, definitely. The amount of tissue required is not actually the based on amount of tissue, but it is amount of the tumor cell content in the tissue. Okay, so for Sanger sequencing, since it has a very low sensitivity, the tumor content has to be at least 20%. But if you're doing it for real-time PCR for a single gene EGFR testing or so, so the amount of tissue required is very less, because it can do it on the 10% tumor content as well. But if you're on NGS, so it requires between 20 to 30%. So yes, the amount of tumor content varies with the different platforms.
So besides the amount of tumor content, I think Chandrakant also
Yeah.
wanted to know the amount of tissue as a whole.
Yeah,
As
yeah. So tissue, yes, definitely, like in lung cancer, the tissue is an issue. So we get to see a very tiny tissue. So I think if the tiny tissue is there, I think we shouldn't go ahead with the tissue.
So when you say tiny tissue,
Yeah,
I'm referring to DNA for NGS.
yeah.
What is the amount of DNA, which is minimum amount of DNA that you will require for Sanger sequencing as opposed to NGS?
So for the Sanger, it will be around 30 nanogram per microliter. But for NGS, you can go with just 2 nanogram per microliter as well.
So Amish says that I'm imposing on his topic. So my sincere apologies to you, Amish. I will stop responding to that. Anuradha has asked a question: do we have a reference genome in Indian population available? The answer is yes and no. If you look at patients of Indian origin in other parts of the world, for instance, in North America and Western Europe, yes, we have that. If you are saying that is it available for Indian population residing in India, I am not sure that we have the same amount of data, but we do have a lot of data. I think Anuradha yourself in Tata Hospital, you have evaluated more than 500 patients.
Yeah.
Not you as a individual, but as an institution, and some amount of data from Indian population is definitely available.
Yeah. So in that regard, I think EGFR mutation, overall mutation rate is around 25%. 31% I felt a little bit high, which is on the south side because it still differs. Like in the south region, we have around 31%, but overall it is around 24 to 25%.
Right. Perfect. Thank you very much. We'll end this session and move on. Chandrakant, over to you.